All right, so yesterday we started talking about gene regulation. The idea is that genes should not always be expressed. They should only be expressed when those proteins that they produce are needed and they're necessary. And we started off talking about how you can regulate gene expression in prokaryotes, so bacteria, because they don't have that many ways to regulate. And one of the primo ways to do that is through what's called the operon. And so I want to talk a little bit more about that and expand on it. So an operon is a whole bunch of genes all together working for one outcome. And that is either to produce a particular protein or to keep that protein from being produced. So to regulate it, in other words. So it's a whole group of genes that are going to regulate whether or not that protein is produced. The one we started with was tryptophan. So TRP, stands for tryptophan. Tryptophan, that word there. Tryptophan is an amino acid. It is an amino acid that bacteria need almost all the time. So it is typically what we would say, refer to as turned on. It's switched on because the amino acid tryptophan is routinely needed, and there's very few instances where it's not needed. Okay, so it's a turned on, or we also call it a repressible operon, and I'll explain why it's repressible here in a minute. So the picture I showed you, and I think we kind of ended on maybe down here. Yeah. Is this guy that shows the entire operon. So it's got all the genes that work together. And I just want to go over that again because I think based on what I saw yesterday and based on the fact that it's new material that you've never seen before, that it's good to do that. So what we want to do, or what we want to control, what we want to regulate, is whether or not these guys here are made. Those are the protein products that will allow us to produce tryptophan. We want to make tryptophan. The bacteria needs it. They need it almost all the time. So those are the subunits that are needed. And they are manufactured from these genes here, these blue genes here. And we know in order to manufacture them, we have to go through transcription and translation. Transcription means that we have to take what enzyme? What enzyme are we going to use? RNA polymerase. So we've got this little teardrop shaped RNA polymerase here, right here. And it is going to bind. Where does it bind? Where does it land? It lands on the promoter. The promoter, yep. On a sequence called the TATA or Tata box. It doesn't start transcribing until it gets to that blue area. But it lands on top of that promoter area. Now, before it gets to the genes that it needs to transcribe, it's got to go across another part of the promoter called the operator, and that's the one that looks like little teeth here. And right now, that's not a problem. We say that it is switched on. It is causing the mRNA to be transcribed, and you're getting these products. You're getting A through E. You're getting those proteins that you need. The reason that is happening is because there is a feedback mechanism that you can use to help control whether or not tryptophan gets needed. And that feedback mechanism is this little thing called the repressor. And the repressor is a protein. And its job is to turn off gene expression, gene transcription, whenever there's enough tryptophan. Okay? So if there is enough tryptophan if there is not enough tryptophan, it will stay inactive. And notice how it's kind of curved here. It has a shape that will not allow it to fit in that operator site. It's a little bit bent. It's not going to fit in there. It's like a puzzle piece. It's all wiggity whack. It's not going to fit. Okay. This is what typically happens with bacteria. They need tryptophan all the time. So normally that repressor protein is not working. Notice how the repressor protein is actually also, we call it upstream of that promoter site. So it's adjacent to the genes that we're going to transcribe to make tryptophan. So it can also be transcribed and made into a repressor protein. And it can either be made into a repressor protein that works properly and fits with the operator, or it can be made into this little curvy one that we have right here where it's not working. Okay. Now that's in the case where it's turned on and tryptophan is not we need tryptophan. Their next slide, though, is now we're just highlighting the relevant bits. So we've taken away some of the other pieces, and we're looking now and we're seeing that now what's going to happen is my little curvy protein is going to bind to tryptophan. The concentration of tryptophan is now so high in the cell that it will bind to this curvy protein. And when it does, it changes its shape. Okay, changes its shape to something that can fit in that operator site. Now, if it can fit in the operator site, this RNA polymerase is blocked. It can land, but it can't get to, it wants to get to over here, but it can't get there without going along that rail. And that rail is blocked by the repressor that's bound to the tryptophan. Sorry? Sure. Yeah. All right, cool. I just wanted to finish that thought for you. Questions on that? Okay, so in general, this is what we call a repressible system or a repressible operon. And it's called repressible because you can turn it off with a repressor protein. But that repressor protein is normally not there. Normally the system is working and it is producing tryptophan. So the repressible operon is one that's usually on. And it's only when a repressor binds does it get shut off. Okay. So the TRP operon is a repressible operon, but there's two types of operons. There's repressible, and there's another one that we haven't talked about yet, and that's called inducible. What if you induce labor? What does that mean? You make it happen, right? You make labor happen. So inducible means you make it happen. So in this case, inducible is usually off. And then there's a molecule called an inducer. Yep, I didn't mean to cross that out. The inducer. And the inducer will cause transcription to be turned on. Now, the key difference between repressible and inducible is that in an inducible system, it is a series of genes that control the production of a protein or protein products that are usually not needed. It's only under rare circumstances or semi-rare circumstances that you would need to turn on that series of genes and produce those proteins. They're usually not needed. They're not necessary. Sometimes you do, but for the most part, you don't. So we want to keep that operon turned off most of the time. And we want to keep it turned off.
In what cases would you need it?
All right, so I will give you an example. Hopefully that won't be too confusing. We're going to talk about what's called the lac operon. Go to the next page here. And a lac operon is an inducible operon. It contains the genes that code for enzymes that are used in the breaking down or hydrolysis. So hydrolysis just means to break it down with water. We know that already. So we're going to break it down. Break down lactose. What's lactose? Milk what?
Fat.
Sugar. It's a milk sugar. Yep. It's a milk sugar. Okay. It's a dimer. It's 2 units long. It's like sucrose, only it's found in milk. Now, a little bacteria is setting out on, we talked yesterday about little bacteria and how it needs to conserve energy. Let's say that my freshmen were in here yesterday and they were sneezing up a storm. They weren't. But they're sneezing up a storm and achoo, little bacteria got sneezed out onto the countertop or onto your tabletop. Oh, you. Back away. That poor little bacteria has to survive until it gets eaten by something else, right? Get picked up. So it's going to try and conserve as much energy as possible. But just in metabolism, just in living, just in performing cell respiration, one of the things it's going to always need is tryptophan. So tryptophan operon is almost always going to be on. However, it has another operon, and it's got lots of operons. We're only talking about 2. It's got another operon to break down lactose. Do you see any milk on your desk?
No.
I hope not. So it has the ability to break down lactose, and it can do that, but it normally is turned off unless there's a situation where it is exposed to lactose. Okay, and lactose is actually going to act as our inducer. Okay, lactose is going to be the inducer. It's going to turn on the system. So if, oh my God, you have chocolate milk. That's amazing. Thank you. Thank you for that. That was perfect. So Lucas is drinking his chocolate milk and he gets a little sloppy and his little straw comes out and it kind of whips everywhere. And now it's on the desk. Now the milk's on the desk and the little bacteria are like, yay me, I have lactose. And now you want to turn on that operon so that you can break down the lactose and you can use it for energy. Okay, but you've got to have those enzymes in place. Okay, does that help? Or just like it's like I'm just gonna remember Doc C shaking and that's the only thing I'm gonna remember. Yeah, that happens a lot, unfortunately. All right, this is a little bit different. So now what's going to happen is a protein is going to. This scenario here is lactose is absent. So now the protein is going to sit up here. So like 98% of the time, that repressor protein is going to sit on the operator, and RNA polymerase is just going to get shut down. Right. So what will happen, and that's when lactose is absent and the operon is shut down. Can't work. Woohoo! However, if now my molecule of lactose is here, right? Spilled my milk, literally. Now it causes it to bend, right? It causes it to be this weirdo shape. And now it can't bind anymore. And that's the 2% of the time where RNA polymerase can then go ahead and produce these 3 products, which are the enzymes that you would need to break down lactose. Okay, that's called inducible because what the product, or not the product, the molecule that is in the environment, in this case lactose, is going to induce or turn on the system. In the case of tryptophan, the product is going to cause it to turn off. Okay, so that's repressible. But in this case, we're turning it on. So it's inducible. Okay. How's that? That make sense?
Yeah.
Woohoo! Okay, so inducible enzymes usually function in a catabolic pathway. Their synthesis is induced by a chemical signal. Repressible, remember so let's talk about that. What's catabolic? Catabolic does what? Yeah, catabolic is going to release, it's going to break down. Catabolic. And anabolic is going to build. It's going to build up. Regulation of both tryptophan and lac operons involves negative control of genes because the operons are switched off by the active form of the repressor. But what you really need to know is that you've got two different outcomes. One's going to turn the system on, one's going to turn it off. But it is a form of regulation. It's a form of feedback. So one of the key things that they want you to understand in this class is the idea of feedback, how you've got either a feedback causing it to turn on in the case of lactose or feedback turning it off in the case of tryptophan. Okay. All right. Sweet. So that's prokaryotes, and that's the long and the short of what you need to know about prokaryotic gene regulation. When do you turn them on? When do you turn them off? Now, we need to talk about gene regulation in eukaryotes because there's a lot more stages and places where you can regulate gene expression to turn on or off the production of a protein. All organisms must regulate which genes are expressed at any given time. Genes are turned on and off in response to signals from their environment and their internal cues as well. So, in other words, if the environment is not hospitable, that can cause a gene regulation to change. And same thing with internal. So if you're sick, for example, gene regulation can change and certain genes might not get expressed because you're sick and you want to put all your energy into getting well in the immune system and you don't want to put it into, say, growing, for example. You don't want to put energy into growth hormones to make you taller at that particular point. You want to get over that flu that you have first. Same thing for environmental. There may be something in the environment that is, maybe it's too cold, maybe it's too not enough water. Plants will regulate their genes as well. And, if it's, really dry out, it's not a good condition to go to flower, for example. You want to make sure that the conditions for flowering is going to take a lot of energy. And the whole point of flowering is eventually to reproduction of the plant. However, you don't want to produce a flower and you don't want it to go to seed if you're not in a good situation. So if the soil is poor or there's not enough sunlight or there's not enough water, all those things could keep that from causing a particular protein to be expressed. So let's talk about the different ways that we can do that. Almost all cells in an organism contain an identical genome. Now, so every cell in your body, doesn't matter if it's skin, liver, heart, lung, any cell in your body contains almost identical cookbook recipe. Has the same chromosomes, has the same DNA, has the same genes. But what's different about them is whether or not the genes within them are produced. And it will be different depending on what kind of cell type you are. So, for example, if you are a brain cell, you're going to produce. I got some seniors in here taking psych, right? What do brain cells? What chemicals do brain cells produce? Going to need this for your AP Psych. Serotonin, dopamine, which is a specific forms of neurotransmitters, right? Cortisol. So these are neurotransmitters. These neurotransmitters are produced by brain cells. Okay, you don't need those if you are I'm trying to think of what doesn't involve the nervous system. It's a little bit hard. You don't need it if you're a fat cell, for example. You don't need, if you're a storage cell for fat, you don't need to produce neurotransmitter, acetylcholine, dopamine, serotonin, etc. You don't need that. So you would want to turn off a large section of the genome because we don't need those genes to be expressed. Abnormalities in gene expression can lead to all sorts of issues, including cancer. So there's some cancers where just gene expression is these particular hormones get produced, and you've suddenly got a growth hormone being produced, and you're producing over and over a cell type that you don't need. We often talk about gene expression as transcription. So if you can control transcription, you control gene expression. But the point of the fact, or the matter of the fact, is that there's actually lots of different, and this is a diagram from your textbook, there's lots of different places that it can be controlled at. And if you've got an mRNA that's already been made, and it's a mature transcript, you want to stop that before it goes to the ribosome and it becomes a protein, because that's going to take a lot of energy to make that protein. So there's going to be steps along the way that we can break down. We can stop the gene from being expressed in terms of where it's at, transcription or gene editing or translation. Yeah.
But if you like want a hormone to start and continue for like years, like if you're like going through puberty or something,
then you can do that too.
And they just like start it and don't stop it until.
Yep. Yeah. And I don't know what exactly the mechanisms would be for turning it off, but that is possible too. Yeah, for sure. So one way to regulate it is at the chromosome level. So if you remember, just kind of shaking some more cobwebs loose, chromosomes are tightly wound pieces of DNA. Chromatin is also a chromosome, but it's unwound. It's unwound. It's not condensed. Some parts of a chromosome can be really condensed, so tightly coiled. If they're really, really tightly coiled like this, there's no way that if I am a little teardrop shape RNA polymerase, I can get into that section of the chromosome and transcribe the mRNA. Okay. So one way of doing that is to modify the DNA so that it's wound very, very tightly. Okay. And in the project that you were doing, you can do that with a methyl group. You can add on methyl groups to the DNA. It causes it to be compacted, really tightly coiled. And then that RNA polymerase isn't going to be able to get in there and do its job. Let me see if... Oh, there it is. Actually, it was on the next page. Okay, so on the opposite of that, you can do what's called acetylate or acetylation. And this is where you add an acetyl group to your section of the DNA. And instead of being very, very tightly wound, now it becomes kind of long and stringy, and okay, you get the idea. And now my RNA polymerase is going to be like, yay, I can get in here and I can make my little copy. You're going to make my little RNA copy. Right? Because now it's strung out and it can get to the promoter site, it can access the start codon, it can transcribe the whole thing and get to the stop codon. That's acetylation. So acetylation is going to make it easier and turn on a section of a chromosome. And like I said before, kind of got the cart before the horse here. If you add on a methyl group, you condense it. You make it very, very tightly coiled and reduces the ability of the RNA polymerase to get in there and do its job. So that would be the easiest way to go about doing that, or stopping a gene from being expressed. But that's not the only way that we can do it. There's other ways too. I think this picture kind of shows it probably better than I can. This is acetylation, and you can see how adding the acetyl groups makes it unwind so that it's easier to access.
Wait, so that's for like turning things on?
Yes, that would be to turn it on because now the RNA can do its polymerase can do its job. Methylation is associated with reduced transcription in some species. DNA methylation can cause long-term inactivation of genes. And that means that it could actually turn it on even like during development. And so particular genes could get turned off very, very early on. And that has some pretty big implications for things like early environment, childhood environment. Because if a piece of DNA gets turned off early on, and it's not, at least with our current technology, reversible, then those proteins might never be made. They've done studies in rats where they have looked at rat pups, the little rat pups that were reared by themselves versus rat pups that were reared with their moms. And they found that their DNA is different, that the DNA becomes methylated, so it adds on extra methyl groups to the pups that have not been exposed to mom. They haven't gotten that nurturing. One of the things that mom does with their rat pups is they groom them. They lick them. They clean them. And those pups that didn't have that nurturing behavior, you see high levels of methylation in the DNA. However, when you let them be with mom, now the DNA of the rats is acetylated, so it's long and stringy, and those genes can be expressed. Yeah, and that's in a mammal, right? I mean, it kind of gives you an idea of what could possibly be, down the road for human beings, or not down the road for human beings, but it could also apply to human beings. That how you're raised, how you're nurtured when you're very young, could be very important. And we see cases of child abuse very early on, or child neglect, and that could have. We know it has impacts, but we're just now kind of figuring out that it could have like long-term impacts in the DNA as well. So this kind of has to do with what we call epigenetic inheritance. Although chromatin modifications do not alter the DNA sequences, they can get passed down. So the DNA is still there, but it becomes methylated to the point where it can no longer be expressed, even when it's passed on to the next generation. And that's called epigenetic inheritance, because you're not changing the DNA per se, you're not changing the code, but you are changing the ability of that DNA to ever be expressed. And so it's the idea that environment can impact your genetics and what gets passed on to the next generation. It kind of goes against and our last unit in here, our second to last unit, is going to be evolution. We're going to talk about evolution. We're going to talk about how Darwin's idea was that you don't evolve in a generation, right? You don't get a trait and then pass it on to the next generation. The quintessential example of that is Lamarck. Lamarck believed that if a giraffe just stretched its neck longer and longer and longer, it would, get stretched out, and that stretching out of that neck, or that, being more flexible, would then get passed on to the next generation of giraffes. And then you'd have longer and longer neck giraffes every generation, just because of that one giraffe reaching for higher and up in the trees. Well, it turns out that that isn't the case. However, he was sort of right, because if there is some methylating or acetylating that happens to different parts of the DNA, and that gets passed on to the next generation, and it was that acetylation or methylation was because of the environment, you really are seeing an evolutionary change from one generation to the next. So it's kind of interesting stuff. We'll come back to that when we get to evolution. Let's see. Already talked about that. Associated with eukaryotic genes are multiple control elements or segments of non-coding DNA that serve as binding sites for transcription factors that help regulate transcription. So these are those transcription factors that I had you drawing in your picture. Let me see if I got an example of that. No, we don't. But basically the idea is that you've got a piece of DNA and you've got this RNA polymerase that's going to be coming along and making a transcription of it. And it is kind of like a train on tracks. It can fall off. It can fall off and not produce the whole mRNA. That can happen. But what you have are these transcription factors, and these transcription factors I probably won't be able to draw very well. But they're like other like I said, not really able to draw it very well. There are other proteins that bind to the promoter area so that when the RNA polymerase attaches to it, it's less likely to fall off. So it keeps the RNA polymerase from derailing or missing the DNA entirely and not doing its job. So if those transcriptions or factors are there, it makes it easier for it to attach properly and produce its mRNA transcript. You don't have that, it can still happen. You can still have the transcription, but it might not be done as efficiently and you might not be able to produce as much of a product. So that's all transcription. General transcription factors are essential for transcription of all protein coding genes. And then also in eukaryotes, there are some transcription factors that are associated just with particular genes to help turn them on and control whether or not they get made. Let me see what's next here. All right. General transcription factors at the promoter. RNA polymerase requires the assistance of additional transcription factors to start transcription. Those are just pretty generic. They're just kind of a scaffold to let any RNA polymerase do its job. A few bind within the Tata box in the promoter. Many bind to proteins, including other transcription factors. So this would be a subtle way to control whether or not a gene gets made quickly or not. Sometimes you want a gene to be produced, but you don't necessarily care how fast it's going to happen. Maybe it's okay if it happens slowly, so you don't want to put a lot of energy into it. So you don't produce those transcription factors. And if you don't produce those transcription factors, it'll still happen, but it just won't be as efficient. And you won't get as much protein outcome as you would have if you had the transcription factors there. Okay. Next thing is post-transcriptional factors. And I think that's a good place to stop. So I'm going to stop there.